Tim Errington here to speak to us today, but we'll just wait maybe a couple more minutes. Yeah, so I think you'll see that transcription has started. Towards the end, we'll have some discussion. So if people want to put questions in the chat as they go, as the talk goes, you're very welcome to do that, or you're very welcome as well just to jump off mute at the end and ask questions. So we sometimes if we have a lot of questions, we find it's more efficient to read them out from the chat, but we'll just sort of see how that goes. Yeah. So I think I will start by introducing our speaker today, Dr. Tim Errington. I'm sure that some people on the call will be aware of Tim and his work, and you'll know that he's a Senior Director of Research at the Center for Open Science in the US that aims to increase openness, integrity, and reproducibility of scientific research, which is, of course, a very, very important goal. So Tim studied, he received a Bachelor of Science in Biology and Chemistry from St. Lawrence University, and then he did a master's degree in Molecular and Cell Biology at the University of California, Berkeley. And then he completed his PhD in microbiology, immunology, and cancer biology from the University of Virginia. And so his current role as Senior Director of Research at the Center of Open Science involves conducting research and collaborating with researchers and different stakeholders across different disciplines on a number of meta-science projects that commonly aim to understand the research project process and evaluate initiatives to increase reproducibility and openness of scientific research. And he's involved in a number of major projects, including the DARPA-supported Systemizing Confidence in Open Research and Evidence, or the SCORE project. But today he's going to be talking to us about his involvement in and leadership of the Reproducibility Project Cancer Biology. And I'm sure that a number of people on the call today will have read some of Tim's publications in eLife, and certainly that's been a project that I've been very, very interested in for a long time. So Tim, thank you very much for agreeing to join us at an early hour where you are in Italy, and we're very much looking forward to your talk. Thank you.
Great. Well, thank you for the invitation, and thanks for that introduction. Let me start sharing my screen again. There we go. Yeah, thank you absolutely for that introduction. And yeah, I'm excited to talk to you about the project that I've been working on or had worked on for about 8 years. I'm going to talk a little bit about that SCORE project that was just introduced, just to put it back into context because it's related. First, disclosures of our funding sources. On the bottom there, you can see the variety of funders that support the work that we do at the Center for Open Science. All right, so let me start just with a brief background of what the center is. I want to put this into context because I think it's very obviously very relevant. So we're a nonprofit based in the U.S. Started in 2013 with this broad mission that you just heard: improving openness, integrity, reproducibility of research. What I like a lot about that is it's a couple things. One is it's agnostic to discipline. Right, you heard my background, but Brian Nozick, for instance, our CEO, is a social psychologist by training, and I love the fact that we have people from a variety of disciplines that work for our mission internally, and then obviously that allows us to engage the community more broadly externally. And we also do it through a variety of mechanisms. We are looking at research, which is I'm going to show you today. Kind of what's the state of the state? What are we dealing with? What are our current practices in terms of conducting and sharing research? We also do a lot of technology to help increase the openness, to kind of integrate with the rest of that open infrastructure system, and work a lot with training and policy and communities to try to see if we can shift those. So I'll hit that at the end, just to, again, try to put this bookmark it around what our mission is. One of the ways that I think also to describe our mission is to actually think a little bit about what our challenges that we're trying to solve, or at least one of the challenges. And to do that, actually, it's best to take a big step back. And for those that have seen talks from COS, you might have actually run into this slide before, but I really do enjoy it because I think it helps understand what we're trying to close. So let me describe this slide and describe the next ones after it. So what we have to think about here, and Robert Merton described this in 1942, is what are we expecting from ourselves as a community of scientists? And Robert Merton described those on the left there as these normative values, right? That science and researchers are about sharing, right? We evaluate research on the merit of the research itself, right? Motivated by knowledge discovery, and obviously are always skeptical, right? Considering all evidence, even if it's against our own work. Versus, these counter norms, right? Which is, it's all about a competition, it's closed, we don't share anything. It's all about where you publish, where your, degree is, where you're working, who you're working with, right? All about this reputational aspect. And then it's all about investing in my career, right? It's like, no. What I'm trying to advance is all that matters, not advancing regardless of what the evidence says. And even though Robert Merton didn't describe it in 1942, I like to think about it, and I think others do, which is it's a little bit of quality over quantity, right? Are we trying to produce the best work we can to try to advance our understanding, or are we just trying to produce lots and lots of work? All right, so like I said, Robert Merton described this in 1942, and I think it's hopefully very relevant today. And there's this great study that was done a while back by Anderson and colleagues that were asking NIH-funded researchers, either early or mid, you can kind of ignore that there, that's just whether they had their first large grant, whether they subscribed to those normative values I just showed you, so whether they believed it themselves. And so what you see here is on that large gray, that 80, 90% is saying yes, they agree that the normative values are more important to what they believe and subscribe to than those counter norms. That hash there, the striped lines, that says that they're equal. And then that tiny little black there on the right, there's a small population that believes those counter norms are actually more important than the norms. So this is not terribly exciting. And it basically just says that what Robert Merton described in 42 still applied to this sample, and I'm sure still applies today. We're actually doing some work on understanding this again ourselves. So then Anderson and colleagues said, well, don't tell me what you believe, tell me what you do. When you actually practice your own research, when you're conducting it, how do these two aspects play with each other? And what they realized is that you see a little bit of creeping there, right? That largely there's alignment between what one believes, what subscribes to, and what one actually does. But you can see there's some acknowledgement there, right? There's some acknowledgement that those counter norms start to creep into the way that we actually conduct our behavior. But by and large, right, like it's not dramatically off. So then Anderson and colleagues said, well, no, don't tell me what you do yourself. Tell me what the people around you do. And it completely flips, right? That people perceive other people's behaviors as not being aligned with these values. And so I think this is actually describes exactly what we're trying to understand when we're really trying to shift and improve the openness and integrity and reproducibility of research, is to kind of help tackle this misalignment between what we want and what is actually going on. And this occurs in our perception, and it occurs in the system around us as well. And I think one thing that to also take from this is that even though that is the challenge, right, which is to see if we can tackle this misalignment, that by and large there is alignment, right? These values do matter to us, right? Openness, sharing, integrity do matter to us. And so the question is, how do we align the system to be able to correct for what we're seeing here? All right, so that's just to put it into context, and part of it is like, what are we trying to correct, right? Sorry, let me just first describe a couple of these barriers that are in the way. One of those is, like I said, those perceived norms, right? That this is the barrier to that correction. Another one is the motivated reasoning, right? It's really easy to rationalize why we're having challenges kind of meeting the ideals that we want to. Obviously, we do this in this kind of account of this system of very minimal accountability. And then we're going to go into this in detail in this project, which is right now, largely what we do is all you can see is what I publish, what comes out of my lab even, right? You don't really get to see what's behind the scenes, and nobody gets to really kind of understand really what I was doing in my research. I think this has a huge implication, especially if you start to go farther and farther into that integrity route and the wrong ways that research at large can go. And obviously we're busy, obviously we have concrete rewards that beat these abstract principles, right? That obviously papers, grants, we're people, we need to be able to make sure we can pay our bills. That beats, the system essentially rewards us for what we want to do, and it beats those principles, right? That yes, we want to be open, yes, we want to share as much as we can, but if I'm not being rewarded for that, it's really hard to pay attention to it. And like I said, I was just hinting at that, we're busy. It's really hard to change our habits with everything that we're doing. And so this is no different, which is if we are brought up and are motivated and are rewarded a certain way, and we ask ourselves to change, we have to recognize that something has to give, and it's going to be really hard to change those behaviors. And that's true for myself. And at least in academic research, that really drives one of the big things, which is the incentive system, right? That a lot of the incentives are driven towards getting it published, right? That's the currency, opposed to really focusing on the quality of the work. And so that's something that we'll have to talk about and think through. And so what I was just getting at before I went to that is what are we actually trying to grapple with, right? Like, how much of a concern is this, and where does that concern lie? And so at least in the space that I'm in, and I'll talk about here in the kind of that preclinical bio space, a lot of it came out in around this same time as that Anderson paper was coming out, 2011, 2012. The big ones were these two white papers that I'm highlighting here, believe it or not, or raised standards, that some of you might be familiar with, or hopefully all of you are, which were these really eye-opening papers that were talking about challenges of the pharmaceutical industry, that this is, and it's all retrospective, of, well, when they're trying to take these exciting findings from the literature and try to actually move that through the pipeline, it fails way more often, a lot more often than that they would have liked, right? At really, really low percentages. The challenge here is that these papers didn't really provide much evidence besides that. I mean, they did it anecdotally, but they didn't actually kind of go into the details. And that wasn't necessarily because they didn't want to, it's because they had a lot of disclosure agreements to not share that information. But I still remember these papers in grad school that left, I think, a lot of wondering of like, well, what are we talking about specifically, right? What are the papers you're talking about? What are the challenges specifically you're talking about? What do you mean by you couldn't replicate? What are the definitions that you're using? And then how do we change it, right? Like, what are the specific information? So again, I think it's being scientists of ourselves around this aspect. And that's the one thing that is the criticism of these papers. And so it's a bit of what spearheaded the project that I'm going to talk to you about, which is, this is telling us there's something wrong. We know that there's consequences if there is something that needs to get adjusted, but we don't quite know what to adjust. And at the same time, a little bit later here, just to put this in context, there are other information, and then this is just one that works out fairly well. And I don't really love the word crisis, but I'll use it here just to kind of put it in context, which is, there's this survey done by Monier Baker, kind of informally. I'm sure you guys have seen this, which is asking researchers, and this is nice in the sense that they did get a good breadth of early and mid and late career researchers and across a lot of disciplines, asking them if there's some issues that they see with reproducibility. And by and large, at this time, they saw that with the researchers they surveyed, that yes, there's concern. There's some reason to have pause, and quite a significant reason to have pause. What I find more interesting is less this headline, is more about the rationale behind it, digging a little bit into the data and realizing that, one, it is across disciplines, and two, more interesting, I think, is that it's not always someone else, that sometimes it's challenges with your own research, right? That you find some exciting finding, you try to move on, you go back, and you can't reproduce your own work. And so that's something to really, I think about in terms of how we advance. Okay, so hopefully I set the stage a little bit. So I'm going to go into this project that started in 2013, started when the center started, when I started working there, and we finished it last year. We published the final papers, kind of capstoning the whole project. And I first want to acknowledge the partners in it. So it wasn't just Center for Open Science. We had great partners at Science Exchange, which is a organization out in Palo Alto, California, that does a lot of fee-for-service, basically matchmaking of CROs or academic shared facility labs. That's essentially where we were able to gather all the technical expertise that was needed to conduct this experiment. And then we also, at the time, they were called the Laura and John Arnold Foundation. Now they're called Arnold Ventures. We had private foundation funding to support this, which I think is still interesting that it took a private foundation to be able to support a project like this. And the last partner is one that we were able to organize ourselves around a little bit into the project, which is the open access journal eLife. And if you're not familiar with them, I highly recommend checking them out too. They're a great organization. And so between all of this, we were able to have an organization, collaborators that were able to conduct this large project. So let me walk you through first some of the details, and then I'll tell you about some of the findings that we had. So first, the approach. So the approach we had, again, we organized this and started it in 2013. That's when most of the information gathering work was occurring. Is we weren't looking and trying to call out specific papers for concern. We were going after kind of similar to what the Bayer Amgen papers were that inspired us, which is these quote unquote high impact preclinical cancer biology papers. So we went a couple years in the past when we started, so 2010 to 12. We cut out the large like kind of omics screen studies, not because there's not something to look there, but because that's a slightly different approach that we wanted to avoid. So no genomics, no proteomics, no high throughput assays, no novel techniques, and also anything that was too difficult to grab reagent or technique that wasn't within the science exchange network, we would exclude as well. So we had a lot of this exclusion up front, but that meant that we were including a lot of your kind of core molecular cell biochemical experiments that are the kind of the bloodline of this type of research. And also, even if a paper, say, was a large genomic screen and then they did a lot of follow-up experiments to quote unquote validate it in, say, an animal model, we would include the animal model even though we wouldn't go after the screen again. And that's because that was the confirmatory research that was being presented, at least confirmatory in the paper. And when we designed this study, we very much thought it was just going to be about, well, what did we find, right? Yeah, it would be great to hear about some of the challenges, but really, like, what's the evidence? That's what everyone was trying to go after. And we were inspired at the time on another project that was going on in psychology that the Center for Open Science ended up actually taking on and running when we formed. But one of the things I'm going to first start with is not what we found in terms of the evidence, but what we found in terms of the process. So those are the two big papers that we wrote at the end, and that's actually what I'm going to talk to you guys through. And I'll start with the challenges. A key feature, though, that we incorporated in this approach and have iterated, and I've thought through this more with others in terms of like how do we best leverage this type of approach going forward, is thinking through not just doing replications and then presenting the evidence, but thinking about how to make that so we can actually advance, hopefully try to advance knowledge. And the concept here is really this pre-commitment concept. And you can see that off on the right. And Brian and I wrote a paper kind of commenting about this approach, which is instead of just finding some finding that one wants to replicate, going off and doing that study design, but not actually engaging with a process that can hold one accountable, you end up getting a replication at the end. Somebody can look at it and always make up a hindsight bias, right? Oh, you didn't do it right. Oh, if you had only considered X, Y, Z. That's going to happen anyways, but to minimize that is to have this pre-commitment stage, which is before we begin any of the experimentation, is to make sure that the methods and the approach that we're going to analyze it and the materials we're going to use are agreed upon beforehand. That we basically have no a priori reason to think it won't succeed. Now, you still get ad hoc rationalization, but now you have to do that not just in contrast to the original findings, but in contrast to the agreed upon design. And so what this is really doing is we incorporated this into the review process of journals. And this is actually a format that we and others advocate for as a supplement to the traditional publication process. So if we think about the traditional publication process, you start with an idea, design what your experiments are, your study, you collect your data, analyze it, write the report, right? Submit that paper to peer review. Hopefully it gets through peer review and you publish it. And all the review is done at not just what you designed, but what you found, right? The actual outcome. This registered report format that basically incorporates pre-commitment, right? Pre-registration. Those are kind of similar words I'm using for the same concept. Has two-stage peer review where you review it at the study design and then you also review it again differently at the report. But that report one is meant not to be the arbitrator of whether you get the paper accepted. That stage 2 peer review is just to hold you accountable. So the stage 1 is actually what allows you to have in principle acceptance. And if you want to know more about this, you can see that link at the bottom. Like I said, there's actually a lot of journals that are doing this, not just for replications, but for quote unquote like novel research, if you want to use that word. So anyways, we incorporated this process with the journal eLife for this project because we felt that it was perfect because it allows us to do a couple things. One, it gives us insight into the study design through the peer review process. And two, it means that eLife will publish our results as long as we follow through, right? That stage 2 peer review of basically saying, did you follow through? Is everything okay? That means they would guarantee publication of the outcomes. And that was important to us because it's really hard still to publish null results or replications. So here's a slide from 2015 of a group at Harvard. Actually, these are some grad students, I think, at the time, who did a really nice infographic. So I still use this because I think it's a wonderful job that they did to try to explain the complex project, and I think it works well. So, and I'll walk you through this a little bit in the next slides with detail. When we started the project, it was, like I said, identifying some papers, some key conclusions, so a subset of the experiments. By key, we usually meant something in the abstract, something that was of drawing of high concern. So usually we try to find something that had an abstract tie to it. we'd start with the paper, right? So we should always start, read the paper, gather, figure out what they actually did in the experimentation of the key figures that we were going to replicate. Usually when we did that, we'd find questions. And so we'd start to document those, compile that information, and then we'd actually reach out and contact the original laboratories, the original authors, and say, oh, can you help us out? Can you help us fill in any gaps or correct any misperceptions that we have in reading the paper methodology or approach? And we'd go back and forth with them, hopefully, and then interface and start to bring in actually the science exchange labs at the same time. So basically trying to iterate over what was done in the original paper as best we can to what we're actually going to do. And it wasn't until after we finished that process that we'd actually then send that information in a registered report, stage 1 format, and send that to eLife. And that's when it would undergo peer review. And there it would go under peer review also with the original authors again. So we kind of engaged original authors twice, one informally through our process and one more quote unquote formally through the peer review process at eLife. Then eLife would bring other experts in and biostatisticians to help us again, not think about what the findings we had, but think about the approach we were going to take for the replication. Again, looking at our methodology, our reagents, our statistical plan, our rationale for sample size, all of that. And only after that was accepted, and then again, we'd have in principle acceptance of the results, did we actually perform our experiments. So we front loaded all of our work essentially. And then when we went to do our experiments, then we were able to do that following the protocol from the registry report. And then publish those findings if we finished it and we're able to have all of our quality control checks as a replication study. So we did this for years, publishing this as best we can and running into some challenges. And I want to talk through those challenges through this process before we get into what we found in those completed experiments. All right, so if you've read this paper, you'll see this. I want to walk through this and give some anecdotal evidence. So when we were first designing this, right, from those 50 plus papers from 2010 to 12, it was around 193 experiments. And I can talk between paper and experiment, but for this, I think experiments makes the most sense for giving you guys some evidence and some numbers behind it. And we kind of ran into some issues along the way. One of those is when we were designing this, we wanted to know what the original data was from the experiments we had. That was a challenge. It was a challenge because very few experiments had open data. That means very few papers had open data. And even if we asked the authors for this, we rarely got raw data. So usually what we would get is nothing, or we would get summary data from the authors. And that basically becomes a challenge. It becomes a challenge that means we have to start making guesses and estimations, right? And so I can do some. If you give me a bar graph and error bars, I can kind of estimate, making assumptions. I don't know the distribution of the data, but I can get some estimate of the variance. If you give me a representative image, then I'm really stuck because all I have is a single image and I don't know anything about the biological variance. So, but again, we can estimate it. So we were able to get past this, but this was a big barrier to us. And especially when you think of the push now for more data sharing across organizations, across continents. I think this really highlights the immediate challenge and opportunity we have in front of us. All right, so that was one challenge. We also wanted to understand the comparisons that were done in the paper. So we can read the paper, obviously, try to infer what the tests are. Sometimes there'd be statistical tests reported, but when they were reported, it wasn't very clear what they were doing. So again, sometimes what I'd see is a bar graph, I'd see an asterisk, and I wouldn't know what that asterisk meant. I guess P less than 0.05, but what test did you do? Sometimes it's not 100% clear what the comparisons were depending on how the lines were drawn. I think you've all seen this before. And it makes it really challenging because it means I've got to kind of infer it myself. And sometimes, right, it's really obvious what the conclusions are of the paper because it's quite dramatic in terms of the difference between, say, two conditions. Other times it's a lot blurrier. Nonetheless, it means I've got to make assumptions. I've got to infer what one is doing. And again, we can get past this, but it's a challenge in understanding the paper. And when they did have tests reported, again, similar to data, we didn't rarely see code shared openly, and after request, we'd rarely see shared code. And here, shared code would be anything, right? An Excel sheet that anything that told me the test that one was doing, Prism files, something like that. Rarely would we get that, and it's because it just wasn't possible to be able to share that information. So again, we'll come back to this in terms of challenges and opportunities in a second. But I can get past all of this, right? We can make assumptions of the data. We can help infer the analysis reported, even if I don't know the exact test. I can redo code as long as I have an understanding of what one is doing, or I can, make my own assumption. So I can get past all of these. But the one that was the biggest challenge is the reagents, the actual materials themselves. And when we're looking through the paper, there's obviously a lot of reagents that are important, but there were some key ones that we were really focusing on. So those would be cell lines, right? Mouse models or human tissue samples, antibodies, and plasmids. Those were like the big ones that we were curious about, because those are really, really hard to get. They kind of would be an impediment to us being able to advance our studies. And what we realized is in the paper that a lot of the times those were not identified either in a repository, which would be ideal to me, or a commercial source. And so that meant like 70% of the time we had to ask for these reagents. And then I guess good news, depending on how you want to look at it, is about 70% of the time people were willing to share it. So we didn't have people willing to share it because they wouldn't respond to us, or they wouldn't share it with us sometimes. And that meant either stopping an experiment or having to remake a reagent or find a new source of a reagent. Just as a side note on this, to give a clear example of what I mean by how challenging this was in our process. Early on, there was one paper we had where all the experiments we identified involved a mouse model that was generated during the course of the study. And it was this, like, I think, triple transgenic knockout mouse. And then they built this mouse, right? It took them obviously a long time to do that. And then they started to characterize that and perform the experiments on it. And so we wanted to pick up the place where they left off. So again, similar to a lot of these, this mouse was something that was made in the lab and was not shared anywhere. It wasn't put into a resource for other researchers to get access to it. The only way to do it was to talk to the authors. We asked them, can we get the mouse so that we can perform these replication experiments? And they refused to share them. They basically told us that if we wanted to do experiments on it, that we'd have to go back and remake the mouse because they were busy doing their own experiments with those animals and they weren't going to share those. So we dropped that paper and moved on, but that I think is a massive challenge, right? The thought of basically as a global community, in order to advance, we have to recreate and remake materials instead of freely sharing them with each other. So that I think really highlights the challenge that we had throughout this project. And then when we go into the papers themselves, is we also needed to make sure that we, like I said, understood what was doing, and it was not clear. And this is a real hard one, right? Which is we always had to ask for clarifications. And part of it is because there's so much details going on. But the thing that sticks out to me is when we coded those based on the degree of clarification, that we had, a fifth of the time that these are incredibly not obvious what was being done in the original paper. And I'll give you some examples of that in the next slide, with the next slides. And so that is the real concern, right? Which is if I have a hard time truly understanding exactly what was done in the experiment, right? It's really hard to be able to both understand what you're doing, let alone replicate and build off of it. And just like reagents, here we can ask for help, and we had challenges in that as well, right? So sometime, about 40% of the time, we had authors that really went above and beyond to help us, really hunting down, dealing with the challenges of finding the original information from their lab, putting us in touch with the people who had left their lab to help us fill in the gaps that we had to make sure we were understanding the paper sufficiently. But about a third of the time, we had authors that were either not willing to respond to us or just told us that they were not going to help us. And that obviously is, I think, a bit of the cultural concern that we have here, which is if we're not providing enough information in the papers, the way that we communicate with each other, this makes it a large challenge to move forward and continue to build on this information that we're sharing with each other. All right, let me tell you that and give you kind of an anecdote, like I was saying. So here's an example of a paper, right? So on the left, there's snippets from the paper. That's the figure from the paper that we were focused on. You can see representative images, you can see bar graphs, you can see your asterisks, pretty classic. And then below that's the material methods section. Very short, very brief, and tons of references. So we would follow those references, right? It says this assay was performed as described elsewhere. So we'd go to that one. That usually would send you to another paper, that would send you to another paper, that would send you to another paper. And eventually you're about 20 years, 30 years in the past, looking at a paper that has nothing to do with the methodology that you're looking for. This is a real problem, right? All of the thought is on what I found, not how I found it. But nonetheless, we can start here, piecing together as best we can what was done and compiling what you see there on the right, which is just a screenshot of like essentially a Word doc that we would compile what we believe what was done, all the questions we had, we'd highlight those and share those with the authors, asking for any help that they could with any of the information that we had, as well as correcting any of the information that we pulled out of their paper in case we got something incorrect or we missed a detail that was hard to find. What were the details? What were the questions we were asking? Like I said, they varied in degree. Sometimes it was not understanding the specific antibodies that were used, right? It'd be like, that's great, I used a MYC antibody. Great, which one? Can you tell us what you used so we know where we're starting from? It doesn't mean that that's the one that we have to use, but it's nice to know where we're starting from so we have some comparison. We lack key details, like what were the sex of the mice, right? We know there'd be mice. There was one paper where we didn't even understand the strain of the mice. We wouldn't know how long treatment periods were for, right? You'd say, great, give a dose, and then you harvest, and we'd have no idea what the treatment period was in between, which I think is a very, very, this is an extreme detail to really understand what was going on in the paper. Wouldn't know the instrument softwares that were used, wouldn't know, a lot of the times it would just be generic protocols. So this is the big challenge we have, right? Which is we want our protocols, we want our methodologies to be open-ended, that the finding is super robust to all of this, yet we know the devil's in the details and we don't share those details. And the same thing goes for the data that was showed. Sometimes the data was quite obvious what the variables were, and sometimes it was completely not obvious with very little detail, right? Like, what's the positive area? Well, how do you define that? How do you measure positive area? These were very ill-defined in a lot of the papers. And a lot of the time, the data would help us, and that's why we'd always be looking and hunting for that. But like I just showed you, we'd rarely get the raw data. And what the responses we'd get back actually is not, yeah, there was some resistance of I don't want to help you, but especially from the ones that were willing to engage with us, I think this actually shows the state of what we're dealing with and how we have to really focus on helping advance. Which is the authors were stuck with some tough spots, so they just weren't prepared for it, or they weren't being rewarded, like I just told you, right? We'd get responses saying, oh, that's great, the details you have are there, but the journal told me to cut down the text. There's one we had where the evidence that we were looking for, kind of getting into the weeds, was in a figure that they had to remove from the paper because they went over the figure limit, right? So these are details that we want to share with each other. We know we should, but now our system of the publications are preventing us from sharing that. So I think that's a real sad thing. And there's an opportunity to get around that we definitely need to work on. Like I said, the data's not there. I definitely heard the equivalent of the dog ate my homework here. There's one where there's somebody that moved on, they were starting their own lab. I think they were a postdoc when they did the paper. Now they're in their own lab. We asked them, oh, can we get access to your data? They said, oh, yes, but I don't have it anymore. Go back to the original lab. That's where the data is, which makes sense. We go back to the original lab. We're saying, oh, can we have access to this data? And they said, no. When that person left, we just erased all their data because we needed that to move on. Right? This is only a couple years out from when that paper was published and the data were just gone because somebody left the lab. Obviously, the other challenge we had is that we move around, right? These papers are done by grad students and postdocs largely that train and move on, and we have a huge distributed system. And so whether that is you moving on to your next step and trying to look back at yourself, or whether you're moving to another institution and trying to leave the paperwork for someone else to pick up in that lab, it's really hard to do that if we're not doing good documentation. And so we had some authors that, like I said, went above and beyond, would hunt down people all over the world to help work with us, and others that basically recognized that that problem was too difficult to take and that that information was just not in the paper and not easily accessible. And that's because the details just weren't there, right? The data wasn't there, the methodology was not there. It was very difficult to be able to piece it back together because it wasn't put in the paper at the time. Okay, but we did finish some. Like I said, we didn't finish nearly as many as we wanted, right? I said we started with 193. You saw that window down. You can probably see the challenges why we had to do it. And we ended up actually going after and conducting 87, of which 50 we completed. Just as a note there about the why, right? We had finite resources, right? That's great. We had funding from the Arnolds. They actually supplemented their funding. We had a lot of donations from a lot of organizations, but we had to be very careful about how to make sure that we were responsible for those resources, and also make sure that we were always doing the highest quality experiments that we could. And so what that meant is this tug of war that I think we all do, but this project really showed it to me pretty cleanly, which is when we run into these challenges where I have to remake something or I have to try to spend all this time regathering information, it's at cost of doing the rest of the work. So again, we tried to focus as much as we can on the quality over the quantity, but definitely was confronted with that throughout the project. And when we did conduct these, right, these are ones that had approval, got information, eLife agreed to publish those. They went through the peer review process and got accepted. we still had some challenges that we ran into, some that are not terribly obvious and some that were just kind of a bit of a disappointment. And one of the disappointment ones was the reagents shared, right? Even though we had information to be able to have access to information, they weren't all actually shared. We'd have people that would not actually go through and sharing that. We'd have to remake them. This would largely be like plasmids that would do this for cell lines. Only one time did we actually decide to do this, to actually intervene at the journal level and get the journal to enforce the policy that they had to share. And it's just because it was way too much. We would have had to drop the entire study at this stage. And it's also tough to do in the sense that it is the right thing to do. That's what policies are for. It's also a great way to really get someone pretty upset with you. But nonetheless, we had challenges even when we were ready to conduct the experiments. And as we all know, things don't always go according to plan. Actually, what is amazing is that a lot of the time they did go according to plan, right? That we were able to actually conduct quite a few experiments exactly the way that they were designed. Again, I'm referring to the quality control, the positive, negative controls working, not the outcome of the experiment. That is what it is. So that's actually wonderful that we had a number of experiments that proceeded the way that we designed them. But a lot of the times we had to do modifications. And some of the times they were quite few. They were kind of minor optimizations, nothing that was overly worrisome, but we did want to make sure that we documented and communicated that we had to modify some of the experimentation. Again, I say that, but if you get a different result, it could be that our modification actually is part of the reason why the experiment didn't replicate or not. Again, devil's in the details, and when we lack details, we're stuck making assumptions. But sometimes, right, we had 17% of this happen in the experiments, we'd have really extreme modifications that had to happen. What I mean by that is like model systems not behaving at all the way that they were intended to work, where we really had to kind of go backwards, always trying to engage the original authors, especially when they were receptive to it. And there were a couple of times, there's one experiment in particular where we just could not figure out the way, including engaging with the authors, to get transduction of cell lines for injection into animals to be at essentially the right level of expression. We weren't sure if it was a plasmid problem. The authors were so generous in terms of like sharing their ideas, resharing reagents with us. And in the end, we kind of just stopped it because we were kind of going round and round in a circle, trying to figure out how to optimize this experiment. And eventually we just stopped because we ran out of resources, right? We just couldn't keep investing in this over and over and over again. And so that would actually kind of decrease the number of experiments as us trying to figure out how to get these model systems to behave. And we were able to implement those modifications most of the time. Like I said, I just gave you an example of one that we couldn't implement because we essentially just stopped it, right? It wasn't obvious what to do and we just couldn't continue to push resources into it collectively as a project. So in the end, we finished 50 experiments. Okay, so here we did, I largely talked about reading papers, to be honest with you. The first handful of the years was really just kind of trying to understand original papers, trying to gather information and trying to see if we could implement that. And what we found is just as a summary, transparency and accessibility were the biggest barriers, right? Not understanding what was done was the largest barrier we had. And that included not just the access to the data behind it, it was the reagents as well. And it highlighted when we operate that way with very little transparency of system, the high dependency of communication, right? The need to be able to communicate with authors and knowing that that's actually also a barrier, right? That we can't always get that information. And it's not necessarily always because someone doesn't want to, it's sometimes because they're not set up for it, right? The system's not set up to make this easy. So those are huge challenges, I think, that actually impeded our ability to understand and replicate, or we could have just as well been trying to extend the research. The same problems are in the way. And when we did do the replications, like I just kind of ended at the end, we ran into challenges in terms of them not always behaving the way one was expecting them to, according to the controls. And like I was just hinting at, that collectively, this led to an inflation of the resource challenge at large, right? That all of these challenges kind of basically increased cost, time, and the uncertainty of what we'd be able to finish. All right, so now I'm going to go into what we found of those 50 that we finished. And before I get into that, I want to talk about how challenging it is to think about how to assess replicability. There's lots of ways to do this from a statistical sense, and we kind of tried to use those as much as possible. I'll go through those, and then I'll get back to one more that we didn't really interrogate ourselves as a team. And this is important because this is, I think, kind of a part of the crux, right? Which is, did it replicate? I get asked that all the time, and I still do. And now I feel like we've come up with at least an answer I can give that's satisfactory to the complexity of this question. And what I mean by the complexity is it really depends what you're looking for. What do you mean by something replicating? So there's lots of ways to do this, and these are just the handful that we looked at, and these are just kind of brief little simple diagrams that kind of illustrate it. So on the left would be success, and on the right would be not success. So you could look at, do I just get an effect in the same direction? Do I see the finding in the same direction, right? So that's really low bar, it's binary, right? It's just saying, is there a signal? You can use your, statistical inference to help you, right? That little asterisk there, which is same direction, statistically significant. That's a pretty common one people like to do in some disciplines. You can look at whether the confidence intervals of the original or the replication are within the opposite, right? So basically taking into account uncertainty and sample size. That's a pretty good one to think about, right? Which is, is it a reasonable assumption to think that they're near each other? I can do another approach called a prediction interval, which is whether it's, you predict the finding. That one's tough because it has some caveats to precision of the original finding. The one that I prefer is actually getting more into just the strength of the effect size relative to each other. I'll highlight that more in a little bit. And then the last one we can do is actually combine them, right? Say, well, we can use meta-analytic approaches to say, let's get the best evidence we can, combine the original and the replication together, and say, did the meta-analysis, how does that look relative to the original finding? So again, you can see success or not success on the bottom. So we basically took all these different approaches. They all have pros, they all have cons, and we looked at each one of those. And depending on what you want, depending on which number you want to choose, you can get anywhere from rosy to not so rosy, or very not rosy to not so rosy maybe. And this is actually where this is the challenge we've had with those Bayer Amgen papers. It's not very clear what they were using, if they were using any of these at all. And a lot of this is about the degree of the scrutiny of that test that we were using, right? So you can get anywhere from 25% to 63%, depending on what approach we use. What we decided to do is not look at these as individual, but to kind of try to aggregate those together, saying, all right, well, these are a bunch of different approaches. We're not sure which one's the best. People can very much make arguments for and against all of them. Instead, let's just ask, of these 5 criteria that we can measure, how often did these experiments, these effects in particular, succeed at them, or how often did they fail, right? So, and we can then bin that into what you see there on the top, which is the degree of how those vary, and then just ask, how often did they succeed 3 or more times, or how often did they fail 3 or more times? And what we see is that collectively, we see that 54% failed on most criteria. As a note, when we were doing these experiments, and I kind of didn't really get into this before, is when we were picking these key experiments, by and large, they would usually be positive findings, right? They'd be treat 2 different animal conditions with drug X and see if that increases tumor-free survival or something like that. So some effectiveness of a treatment or an intervention. But sometimes they also included key null results, right? So if I had a drug I was giving to look at tumor size, maybe I also was looking at any impact on secondary tissue like heart, right? To make sure that I had no noticeable difference there. So that'd be a key null result. And we didn't include those key null results when they came up or were asked of us during peer review, just to make sure that we had a good idea of what we were set out to do in the experimentation and interpretation. And even though the sample sizes are quite different, it is interesting to think that the positive effects were actually even harder to succeed than the null effects. But like I said, that I actually don't find that as interesting. What I find more interesting are the effect sizes. So these are 2 graphs that are just showing a summary of a lot of information. So I'm going to try to walk through this a little bit. And A and B are the same things as B's zoomed in on the little box there, the smaller numbers, so you can get a good appreciation of this. So what you see here on the X is the original effect size, right? So the difference between treatment and control, including the variation. And we'd make every original effect positive by definition. And then the replication effects on the y-axis is relative to the original effect. So you can see this graph now allows us to see kind of how these 2 are interacting with each other. And that diagonal line that's running through it, in that ideal world, everything would be scattered along that diagonal line, right? That if the original effect was small, the replication effect would also be small. If the original effect was large, the replication effect would also be large. And they'd all go up and down that 45-degree angle. And instead what you see is they're all hanging out below it, right? They're all hanging, for the most part, hanging out well below the diagonal line. And on average, we see that the replications were 85% smaller on the effect size compared to the original. And the color coding there just tells you whether the replication was statistically significant by p-value. So this 85% is really crazy when you think about it, right? So if I'm doing an experiment in a mouse model, and let's say I had an original study, and it showed that treatment with some drug or some intervention, increased tumor-free survival by 20 days. That's actually quite reasonable. That's a pretty exciting finding. We in our replication would say we did the same experiment and we saw increased survival by 3 days. Right? Like, that's crazy, right? It's not even close. I think this is really dramatic and something to take pause on, because this is, to me, actually what is exciting about the findings in this area is the size of the effect and the potential of that to translate up the pipeline. Here's another way to look at that, specifically on the effect sizes. So this is looking at all the data on the left, those positive findings I told you about, and those null findings. And we're just looking at the smaller effects for clarity of visualization. Again, you can see the original findings here. We made them all positive by definition. This gives you the distribution. So you can see that you've got a nice distribution of findings up from very small all the way up to these larger 5, the relative unit. And then the replications, it's anchoring way down, right? You can now you can very easily see that 85% drop on average. And then the distribution very much being centered around 0, including some that are obviously going in the opposite direction as well. If you like your p-values, we see the same thing playing out in p-values, right? The original findings, by and large, have p-values less than 0.05. And then those replications that are off on the left side, those all shoot with distributions that are much, much higher, right? So very much suggesting evidence is not similar as the original. And particularly important here is we also decided, because we were trying to understand not just if we were able to find the same results, but if we could explain it. And the tough part is we couldn't really explain it. We could not really explain, looking at a number of different factors from what I was showing you before with challenges and other variables, exactly what was predictive or not of a replication succeeding or not. One of those ones that we were looking at was whether the experiment was an animal or non-animal, right? So that's, I think, fairly straightforward. And that's obviously very important in this space. And there's been a lot of appropriate concern around the complexity of animal experiments and maybe that being a huge concern. And I actually still think that's true. Our evidence didn't really kind of point to it directly. And when you look at one of these, like, remember I told you that whole list of like different ways to look at replication success. If we look at one of them, it sticks out quite dramatically, significant in the same direction, right? You see animal experiments only had 12% of the replications that were significant and in the same direction as the original, compared to non-animal, where it was 54% significant in the same direction. So that was like, that very much stuck out to us as like, oh my gosh, something must be going on. However, if you look at the effect sizes, it's very similar, right? 84, 78%. And so what this suggests to us is that it's not necessarily animal, non-animal per se. There are elements probably there, but it's more about the size of the original effect. If you have a very small original effect size, which you happen to do in animal experiments for the most part, there's a good chance that you're not going to have a high replication rate. And it's largely due probably to small sample size and other variables that are at play. So those were what we looked at, but there's, like I was trying to say before, there's one more that's pretty important that we didn't really get into, and I still actually want to do more of this, which is, I think, another approach is just, what do experts think, right? What are our subjective opinions of replicability? Because in some cases, that actually is probably what matters the most, right? Is trying to distill all of this information to numbers is missing the complexity of the research that's going on and how we interpret it. And instead, we should be asking, what do people think that understand this space? And we were fortunate in the sense that, at least at the paper level, this actually was occurring at eLife the entire time, right? The peer review process of eLife doing 2-stage meant that at that last stage, when they were reviewing our outcomes, and just, again, making sure that we were presenting it correctly, that the editors themselves were actually giving assessments, right? They were summarizing the evidence themselves and kind of putting it in the bins as either positive, yeah, this succeeded, based on how they see fit, that it was mixed, some of the experiments succeeded, some of them didn't, that it was negative, nope, these did not replicate the original findings, or that they felt that it was not interpretable from their end. I'll be honest, I agree mostly with this, but I disagree with some of these in particular. And that actually shows, I think, the beauty of something of subjective assessment is it allows you to see how variable the response is. But I think what you see here as well is that, depending on how you decide to cut this, is that you do see that there is it can be as rosy as you want or not as rosy as you want it to be, depending on how you want to cut this through. But I think this is actually a really interesting way to think about how we assess evidence and essentially what we were able to learn throughout this entire project. And again, this is just on outcome. This is not about challenges. Okay, so let me summarize a little bit and think about what it means to interpret these failures, right? And I think sometimes it's really easy to jump to, oh, the original finding's wrong. It's a false positive. And it could be. That's a completely logical thing. Another one that we could jump to on the opposite side is, oh, the replication's wrong, right? It's a false negative. And that's true as well. I mean, both of those are on the table, despite, especially on the replication side, despite us trying really hard on the front end to minimize this, it's very likely we did something completely wrong. I think probably what's really at play for these, if I'm trying to think without any ill intention, is that they're both right, that the original is right and that the replication is right, because if the experiments were done and the controls work and presented, presumably they're correct. And what we are lacking is we don't actually understand the conditions. That's actually what's happening, that there's always experimental differences between the two, but we don't understand those conditions well enough. And I'll come back to that in a second. Okay, so some conclusions on the meta-analysis. So like I said, replication effects were much weaker, 85%. To me, that's actually, if you ask me the one big take home, like that's the one that's my take home. But I'd be curious to hear what others think. Depending on what criteria you use, like I said, you can have success kind of moving all over the place in terms of a number, if that's what you want. And that's partly due to the liberalness of the test, including subjective. That's a type of test. Like I said, it's interesting to think about, although we have to be cautious how we interpret those positive compared to null results that we replicated. And the same thing, like I was just saying with the animals, right? The animal, non-animal is interesting, but because they have similar declines in magnitudes, we think it's more about the original effect size. But that's an area I think that would be really worth investigating more. The highlight to me is that there's room for improvement. If we think about how successful we are in terms of advancing our agenda and research and moving the enterprise towards helping human health, using this as the part of that pipeline, we're doing amazing things. Yet we're doing it with all of those caveats I just showed you. We're highly inefficient in our system. And I think there's a lot of room to do and some very low-hanging fruit to improve our overall scientific enterprise. And we're not alone here. We're not alone in the sense that what I just showed you is not unique. Right? The challenges are, right? The specifics are, but the issues at large are not, right? The findings are not. And so here's an example showing you, again, don't compare them directly compared to each other because they're different approaches and the details, but what was done in psychology to what I just showed you in preclinical cancer biology. And what you see are fairly similar outcomes, right? That not everything's lining up perfectly, that depending on what you take, you can see findings that are not significant in the same direction, and that you see this regression of the mean. You see the effect sizes dropping and becoming smaller in replications as we attempt to do it one more time. And we can keep going into new disciplines. And actually I wanted to talk about a little bit is that this is something that's going on across disciplines, that if we look at evidence across a lot of the replications that are possible, especially in the social sciences, which I'm showing you here, is that by and large, the evidence does look weaker, right? So here are graphs of different projects, like in psychology or looking at ones in experimental economics, where you see the 0 line there, that dotted line is where the original and the replication would be identical. That's the equivalent of that diagonal line I showed you. Anything above it would suggest that the replication is stronger. Anything below it suggests weaker evidence than the original study. And what you see that is if we look at a lot of collective evidence, looking at a lot of different types of replications, that we see a lot of regression that's coming down. It's hard to sort through what is replicable and what is not, is another way to say this. And when we're assessing it, it's really hard to think through how to do that. Right now, I would say that a lot of the times when we assess it, we go down at the bottom of this scale, right? And these numbers are not here in terms of suggesting that they're accurate. There's just more to make a point, which is we use heuristics right now, right? We're like, oh, what journal is it in? What authors published it? And we do that really fast, right? We can, anybody can do that. We do it at scale, probably do it a lot more than we should for certain key aspects like grant reviews and promotion and tenure. But we know it's very inaccurate, right? We just showed that that's actually not the thing to be looking at potentially. We could do light review or heavy review. That costs a lot and has concern. And help us, right? This is where there's, I think this is where things like peer review are highly helpful. It allows us to do some type of looking, although it's, as we know, lots of bad things still sneak through it. And also it doesn't necessarily adjust, like I said, all of the challenges I just showed you, which again suggests none of these are fraudulent. They're just not presenting evidence transparently. We can redo the analysis, we can redo the replications like I showed you. We can have huge programmatic areas that do this, and those cost hundreds, if not millions of dollars, and takes months, if not years, as I just showed you in this project, right? This project is up on the top. Hopefully the accuracy is high, we'll see, but it took years and took millions of dollars. So how are we gonna sort through assessing evidence, assessing our confidence in it until we get this system kind of to keep in doing it? So we should improve the system. I'll get to there to a second, but I want to just talk about how we still have to be able to sort through, like how much confidence we should have in the research that's being presented, since it is hard to sort through it outside of just doing it yourself. And this is the SCORE project I was just talking about. I'm going to hint on this. I'm going to show you just a couple slides on this, and then I'll pop back to big picture, which is this really starts to ask, and the evidence I just showed you, which starts to say, can we create a more rapid, scalable method for assessing confidence in research claims? Is there another way to do this, right? And I think we're seeing this across the board in terms of how we can sift through the growing knowledge that we're producing. And this is what the SCORE project has been working on. We've been funded to date from the DoD in the US, DARPA, the agency there that we've been working with. We're going to be continuing this project with funding from the Robert Wood Johnson Foundation, actually moving a bit into the health space as we do that as well. So again, anyone who's interested in this, please reach out to me. I'd love to share and get some insights on what we're doing and help. But basically the idea of the program was to see if we could take the empirical evidence that I just showed you, and this was done in the social behavioral space, but the evidence I just showed you that's similar in the preclinical cancer space, and to build tools off of that, leveraging other techniques like human assessments, and hopefully building automated approaches, right, algorithms that are able to score confidence in papers and see if they're able to do it accurately based on human assessment and accurately based on that empirical evidence. Because that, if possible, could provide a scalable solution to assessing it. Now, this wouldn't replace humans, right? This would be a complement. This would allow us to sift through the papers and hopefully understand it. So we're really interested in this. We have some evidence to suggest that it's possible. And I'll show you some that's not from the program, but is on the horizon of some groups that have been working at this space. So these on the left are, again, this is a very similar graph that I showed you for the empirical evidence. That 50% mark kind of gives you a benchmark of probability of success. So anything higher up means that the people or the algorithms on the right, or the market surveys on the left, think that it has a higher chance of replicability. Anything lower means that you think it has a lower chance. And the color coding, black or white, indicates whether the empirical evidence, whether the replication itself succeeded or not. Here they're just using a singular benchmark. So take that with a grain of salt, as I just showed you. And what you see here is that markets and surveys, people on the left, are able to predict it better than chance, right? That the black dots are up higher and the white dots are down lower, which basically means that the humans are able to figure out, I think, what we're capable of, not all the time, to say, oh, I think this could replicate or not. It's essentially what I was just trying to get at a little bit with eLife. On the right there are algorithms, computers saying, all right, well, am I able to predict replication success? And here you can see again that more black dots are up and less are down. So it doesn't suggest it's perfect. The signal still is fairly noisy, but it suggests that there is a way to continue to improve and build on this to see if we can develop a tool that can help us sort through it. I'm going to stop here on this slide, but I'll add one note, which is it's not enough, in my opinion, and this is one of the things that we're working on, to just score something. It's just as important to understand why. Why is something have higher confidence and why does something have lower confidence? Because then that allows it to be informative for both the reader and the producer of that research. So that understandability is really critical. Think of it as a good review. I want my algorithms to give me critical reviews, not just a thumbs up or a thumbs down. All right. Well, that's one approach of what we can do, but what else can we do, right? So there's a lot of challenges, I think, that were highlighted in here, and I'm going to kind of talk about this in two sides. One is what can we do as a social system, right? So as I talked about briefly in the beginning, the incentives are a huge part of this, and that's because we as researchers are not alone. Or even universities are not alone, publishers are not alone, right? That societies, funders, institutions, publishers, the researchers themselves, we're all part of a social system, right? Science is itself a social system and very decentralized for the most part. And that means in order to do any changes, we need that entire system to be able to move together. It's not just on one piece. Every component has a different play, but it's important how we all move together. So that means aligning institutional policies with open science practices. So at least in the US, the National Academy of Science, Engineering, and Math has been doing some work on that. Journal policies, can they align their incentives not about what they publish, but the open practices that we want to be encouraging? So can they align those policies? And that's some work we've been doing with the top guidelines that I'm not talking about, but I'll provide a link in here if you want to look into that more, or I'm happy to put you in touch with folks on our team. Can we actually figure out a different way of assessing researchers, right? If we're assessing researchers based on the number of publications or where those publications are appearing and not on the quality of the publications, we're just adding to this, right? People are busy and the incentives are misaligned. So can we rethink the assessment of how we actually reward and judge researchers and scholarly work? And there's, again, some great groups that are doing that. DORA is one that I'll highlight. And then obviously we need to be able to provide support, right? It's not enough to say, just do this new behavior, but actually providing training on the methods and tools is just as critical, if not essential, to having this happen. And that last one, what we can do is also what we can do ourselves. So if there's any researchers in this audience, especially early career in particular, but just as good to try new things later in your career as well, it's just to try to incorporate some of these practices. There's a lot there. And I'm a big fan of just trying to figure out which ones you want to incorporate and starting there. And the way to think about it, and you can think about it just in terms of data, which is sharing data is not enough, right? That we know there's so much more below that iceberg. And there's a lot of talk around data being fair. We're really talking about metadata, and data management plans are starting to get a little bit more traction. But if you go below the iceberg, there's a lot more there. And to be honest with you, I think that's actually where we really need to be paying just as much attention, because if we have great open data, but we're not sure how we collected it, and we're not sure that the data itself is of high quality, that doesn't really help us. So that's one thing, which is, can we actually work on sharing our data, code, and other digital materials? That was a huge challenge that I ran into in the project. And there's a lot of work. Again, I'll point to the US a little bit as an example, which is the NIH is moving around, not just supporting the domain-specific repositories like for proteomics or for genomics data, but also try to help with the more generalist repository, the data I was largely showing you, right? So, animal data, cell line data, molecular and biochemical data. This is really going to be hard to tackle, but we need to start supporting that long tail of data and getting that more openly available. And one way to do it is to have the infrastructure that enables it. There's a lot of great groups that are doing work here. The reagents themselves, that's a huge barrier. It's actually kind of eye-opening to me too, about the need to really push harder on repositories. Adgene is just one of many. There's a lot of, I think I'm speaking to a group that understands this. I think physical repositories is something that really needs to start to get advocated and pushed a lot more. We tried to deposit what we could in those repositories when we had rights. We leaned on those repositories to help us find new solutions when we couldn't get access to the reagents we needed. And it was the biggest barrier we had. I think the commercial source is just as fine, but I think these repositories for these key reagents is essential. And so if we can all start getting in the habit, it does two things. It provides the resource to others, and it reduces the burden of somebody having to email you and you having to send that. So these repositories are critical to me, similar to the digital repositories, same concept. Also, those protocols, that enigma of the protocols of not having enough information. There's a lot of great tools that are out there. Electronic lab notebooks, there's tons of options that are there. I'm a big fan of making sure that you think about ones that you can open up, ones that essentially when you write your methods, you don't rewrite it. You can rewrite your methods, but you also point to your methods. You just open up your lab notebook and let somebody see all the details that were done. We tried to do that with our own project, and again, happy to talk to anyone if they want to see some examples of what we've done or suggestions on how to incorporate this. And then there's different concepts. This pre-commitment aspect that I was talking about, I think it's really important, especially for replication or any confirmatory research. Anytime I see the words now, I tested or I hypothesized or a priori, I want to start seeing things like a link to something that confirms that it was actually done beforehand. I think we now have a system and a tool in place, both by themselves, pre-registration, or with journals, registry reports, which is what I was talking about. And there's links here, so you can explore those further. I think these are great opportunities to try beginning these practices in your research, and essentially helps you understand a little bit of why you're testing something, and do you understand the conditions to get a given result. So I'm going to end here. I think this is my last slide on a quote. These are a number of quotes I really like, and this is one that's really good to think about. This is back in the 90s. This is the WAVE lab. It's a computational lab out in Stanford University, and they had this great quote talking about how to make their research reproducible. And I highlight in red what I think you can just swap out easily for any given area of research that you're in and anything that we're talking about. And it's just a reminder that when we publish papers, we still do that. We do it for humans, we do it for ourselves, but that's actually not the work that we're doing, right? That's just, it's a postcard. That the actual work was everything that is behind it. It's the data, it's the code, right? It's the materials, it's the reagents. Like that is actually that's the instructions to get to what was found in the paper. So it's not what I found per se, that's important too, but it's how I found it. All right, I will stop there. Like I said, you can find these slides. Feel free. I think you guys are recording this anyways, but if you go to these slides, there's a bunch of resources here to try to help share out information that I talked about during this. And I think I'll stop and we'll take any questions.
Thanks, Tim. That was amazing. So I'm having a bit of echo from myself. We've gone a little bit over time, but I just couldn't stop you because I just I wanted you to keep talking. It was just fascinating. We've got a great question in the chat from Etchem. I'll read it out. So it is really about, there doesn't seem to be a lot of consequences for people publishing things that aren't reproducible. Maybe you could give your thoughts on that, Yeah, are the rewards of a publication in a high impact factor journal outweighing, any risks around reproducibility not being able to be confirmed?
That's a great question. I just glanced at it. I'm sorry about going over. It's morning here, so I'm like completely lost on time. So it's a great question. I think absolutely. I think right now the entire reward system is exactly about like, what are you publishing? Where are you publishing it? Not the quality behind it. And so the accountability it gets back to thinking this social system. So if you can start at different levels. You can start at myself. How do I hold myself accountable? How do I hold my lab accountable? So there's a lot of dynamics you can have there. How do I hold my department accountable? If I'm in a department, how do I get up to my institution? So I think the reason that we don't see enough of it is because it takes an egregious example to have somebody do it. And it's kind of sad to think about that, right? Like there's a blanking on his last name, but there's a great group up in Cambridge. I gave a talk years ago, and his lab has a bunch of principles that they do. And the first reason they try to do things open and reproducible is because they don't want to end up on the news, right? They're like, that's number one. We don't want to be that lab that people are like, oh, look what they did. And that means sometimes making hard decisions. He gave a great story of basically working with the collaborator. Before they do it, they have a policy to reproduce, basically redo the analysis of everything they published. They couldn't, and they did not submit the paper. And that's because the collaborators they had could not find the actual raw data sources, right? It's like what I was just showing you. And they chose as a lab to not pursue it. I think that's amazing, but you don't see that all the time. And the reason why is because no one's going to look, right? No one will find out. I'll get the paper. I'll get my Nature paper. That's what's going to advance my career. So until we start to shift that, I think it's really hard. But you can, and I think it's really great to talk about it at the lab level because that dynamic is actually where it's going to happen the most. That element is going to have the highest impact on this return on investment.
Does anyone, in the last couple of minutes, does anyone who's online have a question, a burning question that they'd like to ask? Please just jump off mute and ask. I guess, I mean, I read a paper not that long ago. It was about, I think it was from, it was talking about how poorer scientific practices just naturally reproduce because they have an advantage. I think it was Paul Smaglick's paper about, I don't know, something about the contagion of poor science. Inherently, if you take time, if you do things more slowly, you're at a disadvantage to the people who are moving fast. That's a tough thing to overcome, I think.
Yeah, no, it is, right. The system is large, and this is like, at this point, it's largely simulation based, which is where the paper comes from, right? Which is you can sit there and recognize that our system right now is very much set up for success with speed at cost of inefficiency, right? So like I said, take the example I just gave you. You see this in other spaces. I won't share my animal model. It's clinical trials. I won't share my data. Why? because I can advance my career faster, but I do it at cost of the rest of the system. So, and then if my mistake, if I make a mistake, and again, I'm hoping that it's not fraudulent behavior, because that's like blows my mind. But if I just have a mistake embedded within my process, because we're humans, it doesn't get caught until way out at the end, because there's nobody else looking around. The system's not transparent. And so absolutely, we reward that as progress at cost of actual progress. And we've seen this in the past. Like there's been some great things. And I think this is the reason the SCORE project's both exciting to me and also a little bit disheartening to me, which is it means we have to sift through it. That we have so much information out there that we're constantly sifting through and asking ourselves, like, okay, that's great. We're producing a huge body of knowledge, but like, which ones can we actually have confidence in? And I don't want to use the word trust, because like I hope that you can trust it all, but like the confidence is going to have to vary. There's huge varying degrees. And until we kind of get to a system where we start to be more diligent, that's going to be really hard. It's also going to be hard to make the value proposition, because when you do simulations, it's a lot harder than like reality. So changing the culture is going to be very, very slow. But I do think, and there's a bunch of other groups that are trying to do this, which is, can we start to see improvements, not on replicability even, but the big hard question is efficiency. Are we seeing bigger gains with a different approach to going about the research process? So fingers crossed that we do that. But I think, even the pandemic has showed us that there's simple things we can do, pros and cons to it. Like sharing is an important thing. If we want to handle a pandemic, let's share more. The truth is, all of our research should be done the same way. It shouldn't take a pandemic to show us that.
Yeah, I think it's quite sobering that you said that, the issues were really not understanding, but transparency and accessibility, which I think a lot of people who have worked in biomedical science would have encountered those things directly.
Yeah.
I think we'll wind up there. It was a wonderful talk. We're going to have it recorded, and I'm going to recommend that every PhD student at the University of Sydney next door listens to your talk. I think it's something that we need to talk about, and there needs to be a much greater awareness of the fact that we're producing information, and we've all got a role to play in ensuring that can make a meaningful difference in the real world for the people that we're doing this for. Thank you so much. Thanks so much for joining us so early, and thanks for a great talk. Thanks for everybody who dialed in.
Yes, thanks everyone.
Thanks, Tim.
Bye.
Thank you. Yeah, that was great, Tim.